Hey everyone, welcome back to another DNA Master tutorial. Today I'm going to be showing you guys how to generate a comparative map using Phamerator. These are useful when comparing phages in the same cluster. These are very nice to put on posters, so if you're going to present your research or something like that, you may want to put a Phamerator map on. They're very common on posters and they're very good at showing similarity and differences amongst genomes of different bacteriophages. So let's say, for example, we'll just type in. Actually, we're gonna sort by cluster first, which you just do by clicking the cluster, and then we're going to scroll down. Let's say till you find your phage. Mine's an AK, I think. AR. Okay, here we go. So here's my phage here, convoy draft, and as you can see, the other AR cluster phages are around it. So let's say, for example, we wanted to compare convoy with Edgar Poe and Humpty Dumpty, and we also want to compare it to a different cluster phage. So we'll just say an AS cluster, we'll say Galaxy. So we're going to hold the control key while we're clicking them. So again, we'll start off with here, we'll hold the control key or the command key on a Mac, and that will allow you to select multiple genomes. Once you have selected all of the genomes that you want, all you have to do is hit the map button and just give it a few seconds. Alright, so this window will pop up. We're going to enlarge this and we'll see this. So let's make this a little bigger. We're going to scroll that out of here and we're going to zoom in just a little bit so we can get a closer look. Alright, so here is the name of the phage. Obviously you'll see Convoy, Edgar Poe, Humpty Dumpty, and Galaxy. And you'll see the genomes here. Again, these are the genes, these little boxes, and the things inside of them are the NCBI conserved domains among here, which give functions for these genes or for these FAMs. So if we were to zoom in, we could just see the functions of these genes and everything like that. When making a comparative map, Phamerator actually color codes certain things, which is very helpful to see amino acid similarity and sequence similarity and things like that. So actually, if you see here, the purple color means very high similarity. So these, as you can see, like the first half of these three genomes are exactly identical, because you see full solid purple shaded regions here with no breaks in it or anything like that, and you can just see for yourself that, you know, these boxes kind of line up with each other and everything like that. But if we go scroll to the end of the genome, we'll see some differences towards the end. The beginning of the genome is where the highly conserved genes are. Those don't generally vary, but in the second half especially, you'll see some differences. So now we're starting to see some breaks in the solid purple bars. So we'll start to see breaks in the solid purple bars here. And pretty much what that means is no sequence or no nucleotide similarity whatsoever between these regions. So we'll see, say, for this break here, we'll zoom in on it a little bit here. There's a slight difference from these two. So this is Convoy and Humpty Dumpty, I believe. So these two genomes have a little bit of a chunk on the beginning of gene 43 that this phage doesn't, and you can see that here. So we'll see this beginning region here, kind of the red box. We'll see it up here as well. But down here, it's not there. So Phamerator actually notices that, and it draws this nice white break between them, notifying us that there is a little bit of nucleotide differences here. So we'll continue on. We'll see highly similar regions here, purple bars, everything's good. And we'll see another break here. We'll see more white, and then in the middle here, we actually see a small bar. It's not purple, but it's a lighter color, and pretty much the different colors mean different E values. So purple means a 0 E value, so identical, obviously, and the different colors just indicate different E values. So if you mouse over them, you can see the actual E value. The closer you get to 0, the closer the color to purple will be. So this is very, very similar, but not identical, this blue region here. And we'll see, obviously, the white means no similarity at all, and we'll keep going to the end. We'll see some more white breaks. In between, we'll actually see a whole new gene here that was added in. This gene here is not in the Convoy genome at all, so that may be something interesting to look at, or, you know, vice versa. This gene is not in these two genomes, so like I said, that may be something interesting to look at. And Phamerator is also useful when identifying novel genes, or, you know, genes that haven't been genes that are just unique to your genome. And like I said, you can notice that by seeing these big gaps towards the end of the genome especially. And lastly, we'll take a look at the non-related phage that we put in from a different cluster. So phage galaxy, as you can see, it's pretty much entirely white except for a small bar here, 4E-23, so some slight similarity between these two nucleotide sequences. But other than that, you can see that this phage is not related to our phages from the AR cluster at all. So you'll see again some more bars here, but like I said, very, very small similarity, and Phamerator makes it easy to compare whichever phages you want with each other. So if you would want to save this whole chart as something to maybe put on a poster, all you have to do is go up here, you can go to File, Save As, and you can save it to your desktop, whatever you want to name it. We'll save it to our desktop and we'll save it. So then when we go back to our desktop here, this is the desktop of the virtual machine, so we're going to have to get this to our regular computer desktop either by emailing it on the virtual machine to our actual machine, just emailing it to yourself and then downloading it on your virtual machine or something. There's probably an easier way, maybe a flash drive would do it, but this is pretty much what you would put on your poster. Maybe you would chop it in half to make it bigger, or cut out certain regions where you just want to stress some information. And that's pretty much how you generate a comparative map. Like I said, Phamerator is very good at that, and I hope you guys enjoyed and learned something. Thanks for watching. Have a good day, guys.